Welcome to Bioscholar. Today's topic is germplasm conservation. I will talk about germplasm, its conservation, and why do we need to conserve them. In the end, I will also explain the germplasm theory. Germplasm is actually a living tissue that can turn into a new plant. It can be a seedling, a seed, a piece of stem, or even a cell that can turn into a whole plant because it contains the information for a species' genetic makeup, and it is a valuable natural resource of plant diversity. Formally, germplasm can be defined as the sum total of all the genes present in a crop and its related species. It is ordinarily represented by a collection of various strains and species. Germplasm provides the raw materials that are used by breeders to develop commercial crop varieties. Germplasm conservation. A species or a population sample of a particular part of its genetic variation can be maintained through conservation. Since germplasm is the basic indispensable ingredient of all conservation programs, thus a great emphasis is placed on the collection, evaluation, and conservation of germplasm. Germplasm conservation is actually the maintenance of germplasm in such a state that minimize the risk of loss, and allows either its direct planting in the field, or its preparation of planting with relative case. It is also known as plant genetic resources conservation. Conventionally, germplasm is conserved as seeds, stored at ambient temperature, low temperature, or ultra-low temperature. Now let's discuss, why do we need to conserve germplasm? Conservation of germplasm or plant genetic resources is necessary for food security and agrobiodiversity. It involves the preservation of the genetic diversity of a particular plant or genetic stock that can be used at any time in the future. It is important to conserve the endangered plants, or else some of the valuable genetic traits present in the existing and primitive plants will be lost. Main crops produce recalcitrant or short-lived seeds. Similarly, in the case of clonal crops, seeds are not the best material to conserve due to the genetic heterogeneity and unknown worth. The genes need to be conserved. The roots and tubers lose viability rapidly. Their storage requires large space, low temperature, and is expensive. In addition, genetically modified materials may sometimes be unstable. Such materials are needed to be conserved intact for future use. Germplasm conservation is essential to maintain biodiversity because it provides a valuable source of compounds to the medical, food, and crop protection industries. It is important to conserve germplasm because many species are at the risk of extinction and much more are threatened and endangered. It maintains the ecosystem by conserving the great plant diversity that maintains various natural interactions between the species. Germplasm conservation also maintains the aesthetic value of the natural ecosystem. Plant genetic resources or germplasm needs to be conserved so that they may be used in crop research and be used as sources of genes for crop improvement. Germplasm theory. It is the concept of the physical basis of heredity, stated by biologist August Weismann in the 19th century. According to his theory, germplasm, which is independent of all other cells of the body, is the essential element of germ cells, and is the hereditary material that is passed from generation to generation. Weismann first proposed this theory in 1883, and later it was published in his treatise Das Keimplasma. But his view contradicted Lamarck's theory of acquired characteristics, which was a prevalent theory of heredity of the time. Although the details of the germplasm theory have been modified, its premise of the continuity of hereditary material is the basis of the modern understanding of the process of physical inheritance. For more concepts, don't forget to subscribe to Bioscholar and also press the bell icon to get timely notification of every new video. Thanks for watching.